So yesterday we slightly discussed regarding the development of frog. So I'll just revise briefly. So we all know that frog is amphibian and its fertilization occurs in the waters. So as usual in frog, the fertilization is external. So ovum is little bit, it is controversial. Some, according to some scientists, it is megalethal, but most of the scientists believe that it is mesolethal egg and it is telolethal, right? So it is covered by plasma membrane, vitelline membrane, and a jelly coat. So having jelly coat as an important feature of frog's ovum. So this is the ovum of the frog. Yeah, this is actual ovum, the nucleus. So the peripheral part is, cortex. Anyway, here this part with the black dots is yolk that is located towards the vegetal pole. And the outermost part is jelly coat. Anyway, so yeah, cortex has two types of granules: dark brown pigment granules and cortical granules. So anyway, endoplasm has nucleus and many white yolk platelets. So having yolk platelets also an important feature of frog's egg. So egg has two colors. One side is black color, that is called animal pole. The nucleus is present towards the animal pole, and the other side is this side, that is yellow in color, that is vegetal pole, having yolk. So after fertilization, the zygote rotates within the vitelline membrane so that the animal pole comes upper. So jelly coat swells immediately after fertilization and increased in thickness. The sperm enters the egg in the animal hemisphere at an angle of 40 degrees from the center of animal pole. After fertilization, the vitelline membrane is lifted off. We discussed already. So now it is called fertilization membrane. So this is a fertilized, egg or zygote, animal pole, so vegetal pole. And this part is very, very important. That is gray crescent, and this total outer covering is jelly coats. So that is swollen. So anyway, so this is vitelline membrane or fertilization membrane. So this is pre-vitelline fluid, right? So after fertilization, the second meiotic division is completed to release the second quadrant. So this is gray crescent. The entry of sperm causes active movement in black pigments of the cortex. On one side, just below the equator, a crescent-like area appears, which is in gray color. So it is called a gray crescent. So it appears opposite to the point of sperm entry. Sperm entry ki exact opposite side lo oka crescentic shape lo gray crescent ane oka area form avutundi. And so this will be the dorsal region. You remember the dorsal region of the future embryo, and the opposite region will be the ventral part of the future embryo. You remember this point. Okay, so the gray crescent area will be the dorsal region of the embryo. Anyway, so due to gray crescent, it produces a bilateral symmetry. So the first cleavage or division bisects, divide the gray crescent exactly in the middle. Okay, and it will become the median axis or median plane of the embryo. So you remember the gray crescent material functions as an organizer. It is an organizer. So that regulates the development. Okay, without which the development is ceased to exist. So as because when it is removed, when it when the gray crescent is removed from the embryo fails to develop further. And at the same time, if when a normal embryo with a gray crescent, okay, is grafted with another gray crescent, okay, so that embryo produces two embryos. So that's why that is the importance of the, you know, the term organism. So there is a scientist called Spemann. Okay, so just I'll write the name. I'll explain his theories later. Spemann. So he did much research regarding the, you know, Spemann did much research regarding the organism. Okay, so in every CSR paper, there will be a question from the research done by Spemann from the developmental biology or embryology. Okay. Anyway, the path of the sperm in egg cortex is called penetration path. After the cortex in the egg, the path of the sperm while reaching the female pronucleus, so that path is copulation path. So then cleavage. So cleavage is nothing but the rapid divisions that occur in the what we call, so in the zygote, so to produce the embryo. So yesterday we discussed it already, but once again, the first one is so vertical and it is called meridional. Okay, so through the central axis. Okay, so this is meridional, and this is first division, and this is second division. Okay, so that produces four, blastomeres, which are equal. And the third one is latitudinal division. So that produces, uh, you know, 8 unequal or 8 blastomeres, from which 4 are smaller, 4 are larger. The smaller are called micromeres. Okay, so the larger are called macromeres. And the uh, one, two, three. Okay, fourth one, fourth and fifth one. Okay, so they are meridional. Okay, so these, okay, fourth and fifth they occur at a time. Okay, so which produce 16 cells. Okay, so out of which 8 are micromeres, 8 are macromeres, and these are meridional, fourth and fifth. And so simply that is considered as fourth division. And the fifth one is double. Here fourth one is double and fifth one is also double. So it is this one. So this one. Okay, and this one. Okay, so fifth cleavage, furrow or cleavage is double. It occurs same time. So that divides the embryo to produce 32 blastomeres, out of which 16 are micromeres and 16 are macromeres. So after this, after 32 cells stage, okay, so the cleavage becomes irregular, rapid, and differential. Okay, so the micromeres are smaller blastomeres. They divide very rapidly. So whereas the macromeres, where the most of the or the yolk is located, so they divide slower or slowly. But micromeres they divide very fast. Okay, so this is the uh, matter. So micromeres divide rapidly, macromeres divide slowly. So until here we discussed yesterday. So then blastulation. So due to continuous cell division, the number of cells is increased in the embryo. Okay. So here the number of cells is increased. So at the same time, internally, so due to detachment of the cells from each other, so they will be restricted towards the periphery. Okay. So due to that, a central cavity is formed in the embryo. Okay, so that central cavity is called blastocele. Okay, a cavity is formed in the embryo to form a blastula. So that cavity is called blastocele. So blastomeres secrete an albuminous fluid into this blastocele. So that fluid is called blastocele jelly. Now that stage of larva or the embryo is called blastula. So the blastomeres layers are called blastoderm. ఓకే సో అంటే క్యావిటీ కాకుండా మిగతా బ్లాస్టోమియస్ లేయర్స్ ఏవైతే ఉంటాయో ఆ పార్ట్ని బ్లాస్టోడర్మ్ అని అంటాం. ఎట్ యానిమల్ పోల్ ఇట్ ఈస్ జస్ట్ 2 cell thick. ఓకే 2 cell thick layer and the sides and the lateral part and floor of the blastula has many layers. Okay, so this blastula is also called as coeloblastula or haloblastula. See, this is a blastula. So it is its BS or vertical section. Here at the animal pole, only 2 layers or 2 cell thick blastoderm is present, and in the lateral and at the floor of the blastula, so the many cell layers are present. Okay, so this cavity is called blastocele, and the fluid which is present secreted by these blastomeres is called blastocele jelly. Okay, so then fate map. So during this separation or this rapid cell division during the blastula, some presumptive areas are formed. We can identify them. Okay, in the blastula itself. Okay, so the macromeres which are present here in this region, the macromeres will become the future endoderm. Okay, they will become future endoderm. Okay, and the notochordal area it lies in front of the macromeres. Okay, so here we can see this one will become the future endoderm, and this is the future notochord. ఇవన్నీ ఫ్లోర్ ఏరియాలో ఉన్నాయి సెల్స్ అని. ఓకే, సో జస్ట్ మనకు అర్థం కావడానికి ఈ డయాగ్రామ్ ఇట్లా ఫేట్ మ్యాప్ అని ఇట్లా వేసుకుంటాం. రైట్, సో హియర్ in front of the endoderm, so this one, this group of cells will become the future notochord. Okay, and then the between the notochord and the endoderm, so there is a group of cell area that is called precaudal plate that also participates in the formation of the notochord. And to either side of the precaudal plate and between the notochord and endoderm, so two bands of mesoderm. See, 2 bands of mesoderm is present. Okay, so you should remember both this part and this notochord part is actually called as chorda mesoderm. Chorda mesoderm. So in the earlier stage, so this is total mesoderm. So that is called chorda mesoderm. So from which mesoderm and notochord are formed. So the notochord is separate later. Okay, so that is cardomesoderm. So this is mesoderm, this is mesoderm, this is also mesoderm, this is also mesoderm. So this total area is actually cardomesoderm. But later, as the development progresses, so the mesoderm or the notochord caudal area is separated from the mesoderm. Anyway, notochord is derived from the mesoderm itself, you remember. And then this total area, which is made up of, micromeres, this total area is actually ectoderm or future ectoderm. Okay, so above the notochord, so this area is actually the ectoderm. So later, the ectoderm, just like notochord from mesoderm, from the ectoderm, the neurectoderm, so which is also part of ectoderm. So the neural plate forming or the central nervous system producing or the neural region of the ectoderm is separated and it will form that is called neurectoderm. This area, this is ventral area. So anyway, this is the VS. This is the blastula internal arrangement. It is in the internal arrangement. It is the internal arrangement of the cells that will form the future various different kinds of primary germinal layers. Okay, so now remember the macromeres will become the endoderm. Okay, so above the endoderm, okay, so the notochord and the precaudal plate is found, which is part of actually the mesoderm. And to either side of the precaudal plate, the mesoderm present in the form of two bands. So above the precaudal plate and two bands of mesoderm, the separated part from the mesoderm, that is notochordal plate or notochordal part, okay, is present. And above the notochordal area, so the ectoderm is present in the earlier period. Later, so some cells are specialized and separated from the ectoderm to form neurectoderm, from which the future brain and spinal cord are formed. So this area is called epidermal ectoderm. Okay, and this is neurectoderm. So that is purely produces the nervous system. So this picture of the blastula, so in which the presumptive future germ layers are seen, so that is called fate map. Okay, so this is also a kind of fate map. So this is BS. So blasters is here. This is the animal pole, and this part is ectoderm that will produce the epidermis, the future, and the extension, the continuation of the ectoderm is neurectoderm. Okay, so in the earlier period, the notochord is actually near the, it is continued, it is continuous with the neurectoderm, and later the precaudal plate. Okay, notochord and precaudal plate cells are present actually peripheral region or the animal pole region. Okay, so before the gastrulation, their position is like this, and here the mesoderm. Okay, so అవతల వైపు కూడా ఉంటుంది mesoderm, and this will become endoderm, animal pole and vegetable. So now we have seen the what we call the fate map of the embryo in the blastulation or in the blast line. So next step in the development is the gastrulation, which is very, very important. Anyway, blastula has one or two cell layer of cells at animal pole. So it is called blastoderm. Blastoderm antam. So this area. Okay, so blastoderm. So during gastrulation, the blastoderm cells arrange in three layers of cells, namely ectoderm, mesoderm, and endoderm. Blastoderm లోని cells ఏవైతే ఉన్నాయో అవి త్రీ జెర్మినల్ లేయర్స్ గా, ఓకే, ప్రైమరీ జెర్మినల్ లేయర్స్ గా అరేంజ్ చేయబడతాయి. ఆ త్రీ లేయర్స్ ని ectoderm, mesoderm, and endoderm అని పిలుస్తాం. సో దీస్ ఆర్ కాల్డ్ ప్రైమరీ జెర్మినల్ లేయర్స్. ఈ మూడింటిని మనం ప్రైమరీ జెర్మినల్ లేయర్స్ అని పిలుస్తాం. So during gaslation, so these are the events that occur during the gaslation. See, if you observe some events, I will show in the diagram. So this is actual position of the, the embryo before the, prior to the gastrulation. This is the blastocoele. Okay, so these are the, the macromeres. Okay, so in this region, this is the animal pole. So in this region, the micromeres are seen. So up to here. So anyway, here what happens, imagination takes. First of all, you remember these areas. So these dots area is an extension of epidermal ectoderm or ectoderm, that is neuroectoderm. After that, notochordal cells. After that, precaudal plate cells. After that, so the endodermal cells. So, and here, these are the part of the ventral mesoderm. There will be dorsal mesoderm, nothing but the notochord. అటువైపు కూడా mesoderm అని ఉంటుంది, ఇటువైపు మాత్రమే కనబడుతుంది. Right. So what happens here? Invagination takes place, particularly at the gray crescent area. Actually, as a narrow groove is found at the gray crescent area. So gradually, this macromeus area will go, or this total layer of cells, okay, so that will be invaginated into the blastocoele or into the embryo. Okay, so gradually, so the invagination is occurred like this. Okay, so due to invagination, the size of the blastocoele is reduced. So now the newly formed cavity is called archenteron, this one. So archenteron. Okay, so here this is due to extension of the neuroectoderm. So the dorsal part is major part is covered by neuroectoderm, and you can see this is the precaudal plate. Okay, and then this is notochord. So it is gradually, so invaginating, the cells are being invaginated. Okay, so this is precaudal plate, this is the notochord, and this is the neuroectoderm, and this is epidermis or ectoderm, and this is mesoderm. Okay, this is decreasing, what we call the blastocoele. Okay, so this is how the imagination changes the structure of the, larva, sorry, embryo. So here two more steps. ఇంకా లోపలికి వెళ్తే ప్రికార్డల్ ప్లేట్ అనేది ఆల్మోస్ట్ బేస్ వచ్చేసింది. సో ఇక్కడ, సో the notochordal cells are notochord, and this is neuroectoderm, and yeah, this is mesoderm. Okay, so this is final, almost final stage of the gastrulation. So here, due to gastrulation, so this part will become ectoderm, this part will become endoderm. Okay, and this part, so this part will become the future, and mesoderm. Anyway, see, now let's see, due to imagination, from below the gray crescent, below the gray crescent. The macromeres move into the embryo to form endoderm. Okay, so to form endoderm, macromeres అనేటువంటి లోపలికి వచ్చిపోయి ఓకే ఆ లేయర్ మొత్తం లోపలికి చుట్టుకోవడం వల్ల దట్ ఈస్ కాల్డ్ ఇమాజినేషన్. సో those cells will become the future endoderm. The newly formed cavity is called archenteron. కొత్తగా ఏర్పడే క్యావిటీ ఆర్కెంట్రాన్ అంటాం. ద ఓపెనింగ్ ఆఫ్ ఆర్కెంట్రాన్ is called blastopore. నేను చూసాను with dorsal, ventral and two lateral blastoporal lips. బ్లాస్టోపోర్ కి మొత్తం 4 లిప్స్ ఉంటాయి. ఓకే డార్సల్ అండ్ వెంట్రల్ లిప్స్ అండ్ 2 lateral blastoporal lips. So a mass of endoderm cells protrude out through the blastopore called jaw plug. సో ఫ్రాగ్ డెవలప్మెంట్ వ్యాస్కులర్ చూసి చెప్పొచ్చు. సో అట్లా ఇమాజినేషన్ అయ్యేటప్పుడు కొన్ని సెల్స్ అనేటువంటివి బయటకు ప్రొట్రూడ్ అవుతాయి అన్నమాట. సో ఇది సో ద ప్రొట్రూడ్ అవుట్ సైడ్. సో దట్ ప్రొట్రూడెడ్ అవుట్ సైడ్ దట్ లుక్స్ లైక్ ఎ ప్లగ్ that closes a particular hole or aperture. So that is jaw plug. The presumptive mesodermal cells, precaudal plate cells, and notochordal cells move towards the blastoporal lips. That is called convergence. Okay, so future lo notochord ga mare cells and mesodermal cells అనేటువంటివి they move towards the blastoporal lips. Okay, so that means ఇట్లా అన్నమాట. సో ఇదంతా ఈ సెల్స్ ఇవన్నీ. సో హియర్ this is notochord cells, precaudal plate cells. So they are present in the dorsal region of the embryo. So they roll into the embryo. Okay, so that is how the imagination started, and rolling of those cells into the embryo is called convergence. And దూరంగా ఉన్న cells అని ఒక దగ్గరికి వచ్చి అక్కడ blastoporal lip దగ్గర ఆగుతాయి. యాక్చువల్లీ ఆఫ్టర్ రీచింగ్ ద లిప్స్ these cells roll over the blastoporal lips and move into the embryo. So that process is called involution. దాన్ని మనం సో ఇన్వల్యూషన్ అని పిలుస్తాం. The involuted cells move away from each other from blastoporal lips area to reach their respective areas. దాన్ని డైవర్జెన్స్ అంటాం. అంటే blastoporal lips దగ్గరికి ఏ సెల్స్ అయితే వచ్చినాయో సో they will move away from each other because their future sites may be different. సో అట్లా డిఫరెంట్ సెల్స్ డిఫరెంట్ డైరెక్షన్ లో వెళ్ళడానికి డైవర్జెన్స్ అంటాం. The precaudal and notochordal cells are arranged as a plate in the mid-dorsal roof of archenteron. Okay, so precaudal and notochordal cells, they are arranged as a plate in the mid-dorsal. So mid-dorsal roof of archenteron. So here notochordal cells plus mesoderm is called as chorda mesoderm. Right, above notochord పైన by extension neurectoderm cells form neural plate. So above notochord పైన by extension neurectoderm cells form neural plate. So this one. Okay, so that is neurectoderm. So the epidermal notochord plus mesoderm called chorda mesoderm. the epidermal ectoderm extends all over the embryo. Monane Japan. So that is epipoly. Bahiravana Prakriya. So epidermal cells ఏవైతే ఎంబ్రియోకి పెరిఫెరల్ రీజియన్ ఉన్నాయో ఓకే సో అవి ఎంబ్రియో వీపు లాంటి పార్ట్ మొత్తం అవి ఆక్యుపై చేస్తాయి. సో దట్ ఫినామినా ఇస్ నోన్ as epiboly. Okay, but neuroectoderm cells and blastoporal cells, they do not move. Okay, so by constriction అనేటువంటి ఒక ప్రాసెస్ ద్వారా the size of blastopore, so by constriction the size of blastopore gradually decreased. Okay, అంటే బ్లాస్టోపోర్ యొక్క ఓపెనింగ్ సైజ్ అనేది ఆ రంధ్రం సైజ్ అనేది తగ్గుతుంటుంది డ్యూ టు కన్స్ట్రక్షన్ ఆఫ్ ది different blastomeres. The gastrula rotates within the fertilization membrane in a horizontal axis. Okay, so this brings the blastopore to vegetal pole. దీనివల్ల blastopore అనేది కింది వైపు అంటే okay, so this is blastopore, this is archipelago. Okay, so here so this size is gradually decreasing. So and this is the arrangement. Okay, so this is animal pole, this is vegetal pole. Okay, so anyway, so now there is a term called neurula. The neural plate will become neural tube that ultimately forms brain and spinal cord. The neural plate ఏదైతే మనం చూసినాం ఇందాక. So here, this is neuroectoderm. Neuroectoderm size అనేది increase అవుతుంది ఇక్కడ. So this part will become the brain and spinal cord of the embryo. Okay, so దాని తర్వాత ఏముంది? So this is okay, so that is notochordal area and this is precaudal plate. Okay, so and what is this? Endoderm. And after more development, so this mesoderm, it reaches on both sides and the blastocyst is will almost will be closed. Okay, and okay, so that is how now if you observe, this will be ectoderm and this will be endoderm, and this one which is trying to enter so will be so the mesoderm. Anyway, yeah, here also, so this part epidermal ectoderm or ectoderm, so this is neuroectoderm, and this bigger cell mass is the actual endoderm. Okay, so then here gradually size of the mesoderm is increasing. Okay, so here only one side is shown, so both sides the mesoderm occupies, so gradually it will settle between the endoderm and the ectoderm. So anyway, presumptive mesodermal cells, precaudal plate cells, and notochordal cells move towards the blastoporal lips called convergence. Okay, so future lo కావాల్సినటువంటి mesodermal cells, precaudal plate cells, notochordal cells move towards the blastopore lips called convergence. So ఈ ఏరియాకి ఆ యొక్క సెల్స్ అనేటువంటి రీచ్ అవుతాయి. After reaching the lips, these cells roll over the blastoporal lips and move into the embryo called involution. The involuted cells move away from each other from blastoporal lips area to రీచ్ ద రెస్పెక్టివ్ ఏరియాస్ కాల్డ్ డైవర్జెన్స్. లోపలికి వెళ్ళాక అవి వాటి వాటి పొజిషన్కి వెళ్తాయి. సో ఒకదాని ఒకటి దూరం అయితే కాబట్టి దాన్ని డైవర్జెన్స్ అంటాం. The precaudal and notocaudal cells are arranged as a plate in the mid-dorsal roof of Arpentera. ఓకే, ప్రికార్డల్ ప్లేట్, నోటోకార్డల్ సెల్స్ అనేటువంటివి ఒక ప్లేట్ లాగా. ఓకే, సో మిడ్ డార్సల్ రూఫ్. ఓకే, సో మిడ్ డార్సల్ రూఫ్ పైన. ఓకే, సో ఆఫ్ ఆర్కిడ్రా దే ఆర్ అరేంజ్డ్ లైక్ ఎ ప్లేట్. సో నోటోకార్డ్ cells plus mesoderm equal to carda mesoderm. Above the notocard, by extension, neurectoderm cells అనేవి ఉంటాయి. బై ఎక్స్టెన్షన్ అనే ప్రాసెస్ ద్వారా ఫార్మ్ న్యూరల్ ప్లేట్. ఓకే, ఫ్యూచర్ లో నెక్స్ట్ స్టెప్. ఓకే, సో నోటోకార్డ్ పైన ఓకే సెల్స్ అనేటువంటివి సో బై ఎక్స్టెన్షన్ అనే ప్రాసెస్ ద్వారా ఓకే సో ది neurectoderm cells form neural plate. Okay, the epidermal ectoderm by a process called epipoly extends all over the embryo, so except neurectoderm and blastoderm. అది మొత్తం outer cover మొత్తం epidermal ectoderm cells they move. Okay, so by constriction the size of the blastophore gradually decreases. Gas rotates with the fertilization. Anyway, neurula. The neural plate will become neural tube and that ultimately forms brain or spinal cord. This stage is called neurula. సో ఇప్పుడు మనకు క్లియర్ గా ఒక న్యూరులా స్టేజ్ అనేది కనబడుతుంది. సో మీకు బయట వైపు ఎపిబోలి వల్ల ఎపిడర్మల్ ఎక్టోడర్మ్ సెల్స్ అన్ని కూడా బయట వైపు మిగిలిపోతుంది. ఓకే, అండ్ దిస్ వన్ ఈజ్ మేడప్ ఆఫ్ ది macromeres are endodermal cells, so that will become the arpentera or the future gut. Okay, so here this middle part, if you observe here, this middle part, so here this middle part is called mesoderm, that is present exactly between the ectoderm and endoderm. So here the small split, okay, that occurs in the mesoderm, so to form two layers. Okay, so inner splanchnic mesoderm, so the outer somatic mesoderm. Okay, so this is the structure of the neurula. So here you can observe this is also part of epidermal ectoderm, so which is become the these two layers are fused to form a neural plate or the future neural duct. So that is what we call the neurula. So this is the notochord. So fate of germ layers. Okay, here we have now this is ectoderm, this is mesoderm, this is endoderm. So వీటిని మనం ఏమంటున్నాం, 3 primary layers అంటున్నాం, ectoderm, mesoderm, endoderm. సో వీటి నుంచి సో ఏం ఫామ్ అవుతుంది, ఆర్గనోజెనిసిస్ ఇస్ అకర్డ్. ఓకే, ఫ్రమ్ 3 germinal layers. Okay, so here the ectoderm, what does it produce? Right, the ectoderm, so it produces number 1 epidermis ని ప్రొడ్యూస్ చేస్తుంది, cutaneous glands ని ప్రొడ్యూస్ చేస్తుంది, ఐ లెన్స్, కార్నియా, రెటినా, కంజక్టివా. Okay, so They are all produced from the ectoderm. Central nervous system, brain and spinal cord. So the pineal gland, pituitary gland, enamel of teeth. Okay. Okay, so these are the organs or the structures that are derived from the ectoderm. This is very, very important by heart చేయండి. From the mesoderm, ఏమేమి ఏర్పడతాయి mesoderm నుంచి? Notochord, pericardium, peritoneum means so the covering surrounding the lungs and kidneys and the ovary are the testes. Okay, so pericardium and peritoneum. Muscular system is produced from the mesoderm. Skeleton is produced from the mesoderm. Okay, so connective tissue, blood produced from the mesoderm. Adipose tissue. Okay, so and dermis of the skin. Dermis and the second layer of the skin. So here you remember visceral organs are derived from the primary. So anyway, from the mesoderm, notochord, pericardium, peritoneum, muscles, skeleton, connective tissues like blood, lymph, okay, and adipose tissue. Dermis of the skin means second layer of the skin. Okay, so first layer is epidermis. So second layer is dermis. And the visceral organs, most of the visceral organs are derived from the primary mesoderm layers. Okay, so endoderm. So here the internal epithelium of the digestive tract is produced from the endoderm. So respiratory tracts, eustachian tubes, gastric and intestinal glands, so liver and pancreas, so bile and pancreatic ducts, lining of urinary bladder. So derived from the primary endoderm layer. So ఇవి మీరు గుర్తుంచుకుంటే బై హార్ట్ చేస్తే బెటర్. ఓకే, సో లేదా గుర్తుంచుకున్నా పర్లేదు. సో దెర్ ఇస్ మోర్ ఛాన్స్ asking question from this part. Even this also relates to higher vertebrates like reptiles, aves, and mammals also. Okay, so then after gastrulation, so this is the gastrulation is still that occurs within the the fertilization membrane or the egg. Okay, so in frog, within 6 days after fertilization తర్వాత 6 రోజుల లోపల the egg hatches in 6 days లోపల into a larva called tadpole larva. Okay, so tadpole larva అనేది so that is produced. Okay, so it will be, there are different stages of tadpole larva. So one is 4 mm tadpole larva. So and tadpole larva generally the frog is carnivorous, whereas tadpole larva is herbivorous, and it rapidly or voraciously eats the grass and herbivorous material. Okay, so first stage of the tadpole larva is 4 mm tadpole larva. So second stage of the tadpole larva is 8 mm tadpole larva. So this is also known as, famously known as, external gill slit stage. And the third stage is 11 mm tadpole or internal gill slit stage. Okay, internal gill slit stage. So first external gill slit stage అనేది ఫామ్ అవుతుంది టాడపోల్ లార్వాలో. సో నెక్స్ట్ ఇంటర్నల్ గిల్ స్లిట్స్ అనేటువంటి ఫామ్ అవుతాయి డ్యూ టు ఫార్మేషన్ ఆఫ్ internal gills and operculum surrounding the internal gills, just like fish. And an interesting fact about the uh, tadpole larva. So its trunk is almost transparent and it appearance is like a fish. Okay, so anyway, here the third stage of tadpole larva is internal gill stage. Okay, so yeah, the alimentary canal ఎలా ఉంటుందంటే విత్ ఇన్ ది visceral area. So it looks like a clock spring. Okay, so కాన్సెంట్రిక్ సర్కిల్స్ లా ఉన్నటువంటి ఒక స్ప్రింగ్ లాగా ఉంటుంది ఇన్ ఎర్లీ స్టేజ్ ఆఫ్ ది టాడ్పోల్. Okay, so later it gradually decrease in size. It will become straightened mostly. Okay, so small intestine develop well. Okay, during the metamorphosis. Anyway, so the tadpole has 4 mm, 8 mm, 11 mm tadpole stages. 8 mm is called external gill slit stage. 11 mm tadpole is called internal gill slit stage. So anyway, within 2.5 to 3 months. Okay, so దాదాపు 2 to 3 months లోపల larva is metamorphosed into adult. Okay, and this metamorphosis process takes place a month. దాదాపు ఒక నెల రోజులు టైం పడుతుంది లార్వా అనేది సో అడల్ట్ కావాలి. సో అడల్ట్ ఎలా ఉంటుంది అని అంటే ఫస్ట్ ఆఫ్ ఆల్ ద లార్వా ఈజ్ హెర్బివోరస్ వెరాజ్ అడల్ట్ ఈజ్ కార్నివోరస్. లార్వా respires with the internal gills or external gills, whereas the adult respires by skin and what we call the pulmonary sacs or the what we call lungs. Okay, so larva contains fins. Okay, they are disappeared and in their place fore and hind limbs are developed. The tail will be lost. So when the tail is completely lost, then that will become an adult frog. Okay, so there should be diagram. Actually, see, this is the blastopore. Gradually, its size is decreasing. See the diagrams. This is the blastopore. So almost, at the end of the gastrulation, this blastopore is almost, closed slightly by epidermal overgrowth. Okay, so it is almost disappeared. Anyway, so this blastopore will become the larval or adult's anus, and this area will become opposite to this blastopore. So this area will become the mouth. Okay, so anyway, so that's why these are called deuterostomes. So this is your plug. So this is neural plate. This is notochord, and this is precaudal plate. And this is the endoderm cells or macromeres. and this black one is ectoderm or epidermis, and this one is mesoderm. This is the photo of the diagram of the gas flow. So anyway, there should be just wait, diagram of. So this is the diagram of life history of frog. So here fertilization is going on. Okay, so this is how the develop blastocyst, blastopore. Okay, so this is gastrula. So then larva. So 4 mm, 8 mm, 11 mm. Okay, so now the adult. Anyway, so this is the development of frog. Okay, let's continue tomorrow. Mostly I take the class tomorrow also. Chuda, right?